Our next speaker is Joan Merril, and a woman who needs no introduction, and she's going to talk on the update of treatments in development for lupus. Joan.
I'm going to paraphrase Jill Bion, who made the famous statement a number of years ago: the trouble with drug development in lupus is lupus. Now, this is an incredibly heterogeneous disease, and we alluded to that before, and I know that clinically everybody knows just how you can see 3 patients and they may have overlapping features, but each one is quite unique. And then suddenly patient number 1, who had nothing in common with patient number 3, develops overlapping features with patient number 3. And we go, why is this happening? Why are these patients, why is there this moving target in the immune system with these patients and so unpredictable? And I think that many of you have probably seen this slide before because I show it all the time, but it's my talisman. I really think that the person who explained lupus to me best was Chandra Mohan, who works in mice. And so for all the bad mouthing that I do of mouse doctors, you know, by the way, Dr. Hughes pointed out that he doesn't necessarily think that criteria for the antiphospholipid syndrome are all that useful to a clinician. I really want to second his opinion and point out that particularly in that field, the majority of people making up the criteria are mouse doctors. So I've done my bad mouthing. Because our patients really don't read the medical textbook before they become ill. It's quite annoying of them. But Chandra Mohan pointed out, using a mouse model in a very simple and elegant series of experiments, what we're really dealing with. And although a mouse is not a human and a mouse is an overly simplified situation to study because if nothing else, you can tell the mouse who to marry. The complexity within that simple system, and then imagine magnifying it 100 times for the human, really illustrates, I think, what it is we're facing in our clinics. And so he took the NZM2410, otherwise known as a lupus-prone mouse, and presumed that there may be multiple genes even in the mouse itself who's been inbred, and theoretically you could come up with one gene to cause lupus, but he presumed there would be more than one gene in that mouse that was predisposing to lupus. And he bred that mouse to a lupus-resistant strain of mice that, you know, even if you give them pristane and things, they're a little bit more resistant to getting lupus. They seem to have a nice genetic profile that even if you push them a little, they don't really get lupus that easily. And in the offspring, he was able to sort out 3 different types of children, of these mice. And the most important thing about them was that none of them developed clinical features of lupus. I don't know what you call a clinical feature of lupus in a mouse, but how he looked at it was that the mouse he called SLE1 made characteristic autoantibodies of lupus mice, such as antibodies to double-stranded DNA, and it did not get lupus. SLE2 showed evidence of hair-triggered B-cell proliferation and did not get lupus. And these are characteristics we do see in lupus patients. And SLE3 had T-cell abnormalities and abnormalities in the cleanup system for the immune system, in the garbage disposal system, and apoptosis and clearance. And this mouse did not get lupus. So each of these baby mice had some features that we see in human lupus and in mouse lupus, and they did not develop the clinical features characteristic of the lupus-prone mouse. So of course, the next step after that was to breed them to each other and see what it takes to get lupus. And so when he put SLE2 and 3 together, you didn't get lupus. So it looked like, you know, SLE1 was important, and yet we already knew that SLE1 wouldn't cause lupus by itself. So it had to be some combination. So when you put SLE1 and 3 together, you got about 50% fatal lupus, made you think, well, we're almost there. Maybe 2 will be even worse, or maybe it'll have 50%, but it didn't. If you put SLE1 and 2 together, you only got 15% fatal lupus, which wasn't so bad. The risk was not so high. And so that made 2 look like maybe it was a pretty wussy contributor to the situation, and maybe because of the vicissitudes of experiments, 50% was the highest we were going to get it to risk to make it look like the parent mouse. But no, in fact, in reproducible experiments, when you put 1, 2, and 3 back together, you went from the 50% fatal lupus with only 1 in 3 all the way up to 90% fatal lupus, which made it look very much like the parent. And so 2 could be an important set of genes, but it has to be along with certain other features in order to cause the risk. Now this to me explains what we're dealing with, except it isn't 3 genes, it's probably now hundreds of genes. And we don't know how many you need, or how many you need if it's these genes and how many you need if it's those genes, but it's how they're put together in a patient who inherits certain polymorphism or alleles from their mom and certain from their dad, and how it all falls together in a very complex manner is what's giving us lupus. And that is why we face such heterogeneity and unpredictability in our clinic. And so there have been huge challenges to mounting successful lupus clinical trials. And I know that I am very well known for whining about this. In fact, I've made a career whining about lupus trials. And I'm always, quoted as refusing to say that the trial didn't work when it didn't meet any of its primary or secondary outcomes. And please understand that that is not what I've ever said. If a trial does not meet its primary or secondary outcomes, there is no evidence that the drug works. 0. Because we can stand up there and show exploratory analysis where subset 1 and subset 2 did work, after massaging the data interminably, and that proves nothing because we've broken a statistical law called Bonferroni's law, and by random, something was going to work if we looked at enough questions. One of them is going to come up the way we want it. And so it proves nothing when you do these exploratory analyses. And so why am I now going to spend an hour showing you exploratory analyses? Because it can help us generate hypotheses about how to design the next trial. And then if you design that next trial according to one of your hypotheses and it doesn't work, you can be pretty sure that drug doesn't work. But if it does work, you can begin to suspect the drug might work, and you can begin to do more trials of more drugs using those strategies. So the purpose of my whining, everyone, is not to suggest that these failed trials are really great drugs. It is to suggest that we need to redesign trials until we're satisfied that we're looking at a situation with this heterogeneous, complicated population where we can sort them out in some way so that we'll know when we get the data how to interpret it. And so I am dissatisfied with our past trials. We've had a horrible learning curve. And even the Benlysta trial, which I'll show you a little data on in a while, only had such a small treatment effect size that it's sort of disappointing, even though it may be the first drug approved for lupus. And so what are our challenges? What have we been identifying that seem to be in our way? Well, first of all, we don't have good tools to sort our patients biologically. I was just talking to a bunch of people, and it keeps coming up that most of us are using certain biomarkers because in clinical practice we figured out that they work. And one is an antibody to double-stranded DNA. But on a good day in your clinic, Dr. Hughes, how many of your patients have an antibody to, what percentage have an antibody to double-stranded DNA? Yeah, well, that's what I'm saying. It's not all of our patients. And also it depends on what kind of test you use. If you use a FAR, you don't get as many as if you do an ELISA, et cetera, et cetera. And how do we interpret that? So we need biologic tests that are specific to targeted treatments because it might help us select which of the patients are real candidates for that target. Let's assume, given the heterogeneity we just talked about, that everyone is not mouse 1, 2, 3. Some of them are mice 1, 7, 18. Some of them are mice 2, 3, 18. You see what I'm getting at? And if I want to use a treatment that targets one molecule on the surface of one cell, I guess I'd like to know whether this is a patient who needs that